let's go back at 500 years. The toxicologists, and hopefully I don't offend anyone here, claim Paracelsus as the first toxicologist. I'll claim him as the first clinical pharmacologist. And I don't speak German, and I'm not even going to try. So basically, he says dose is important. Everything's a poison, it's just how much you give. So you can poison yourself with water if you drink enough, right? Coming back to someone who unfortunately we lost this year, James Black. This is his Nobel laureate speech, and I was trying to get a really pithy comment, which I couldn't find. So this is like the last paragraph or so of his Nobel speech, and I even had to look up verisimilitude because I'm not that brilliant at English. Anyway, but I'll paraphrase what he says here. You need good translational models. This guy may be less familiar to many of you. In the PKPD world, he's kind of a god of some description. Lou Shiner, who died a few years ago after teaching an advanced PKPD workshop. Learning versus confirming. Many companies, I think, espouse this kind of principle. It's basic science, really. It's basically about inductive reasoning. He actually pinched his ideas. This is Lou. He actually pinched his ideas from an English statistician who works in the States. Called George Box, but it's basically basic science. It's like you use the inductive, deductive loops to kind of make decisions. And actually, we were talking before about linearizing the process. The problem of this kind of process is you can't write a Gantt chart that goes from A to B to C to D to E to filing to lots of money, because actually you have to do some inductive reasoning. And it isn't the case that if you check you've just put 50 studies, or how many it is, into a file, send it to the FDA, you'll get approval and make lots of money. It doesn't work like that. You kind of have to understand what you're doing. So Lou, and this is actually quite a long time ago, this is CPT, more than 10 years ago. You need an organisation that's able to do that. And actually, we still, almost always, as soon as we get into the clinic, we're in confirmatory mode. This drug will work. A colleague of mine in the States has a brilliant phrase that we have this conspiracy of optimism in our industry where the folk, who from the early parts of the discovery process kind of believe in the target, and they kind of have to. I mean, it's not surprising. It's their baby, and they've nurtured this baby, and they've got it so far along, and it's going to go to the clinic. It's going to work. Of course it's going to work. But we're not objective enough. We don't do the learning piece before we do the confirming. We presume it'll work, and then we take all the data that supports that hypothesis and say, yeah, look, it's going to work, and we ignore everything that says, it's not. We just ignore it, right? Push it to one side, because actually we'll run the next experiment, and actually the data will turn out the way we hope. So we have this thing, which can't kind of see the red very well, but the 3 pillars. So it's interesting, actually, other industries use this kind of approach, but it's kind of what I just said before. It's actually just back to basics. If you want a drug to have some chance of surviving and being efficacious in a proof of concept type study, our analysis says, well, actually, you need to get the drug to the target site of action, and actually over a reasonable period as well. So if you've got really ropey PK that says the drug just disappears in an hour, and you want a 24-hour kind of coverage, then but you need to get the drug to the target. And then you need to occupy the target at an appropriate level. So if you're antagonist, probably you need kind of 90% receptor occupancy, something like that maybe. If you're an agonist, maybe you need a lot less. But you need to think about what that target occupancy is. And of course, then you want to have some kind of functional consequence of that. And if you've got all of those, our analysis says you're doing all right. So in a normal, this is a fairly typical Pfizer thing of doing a 2x2 grid. We can kind of look at high confidence up here where you've demonstrated data to support all those 3 pillars, and actually you've got a fairly you've got a reasonable confidence in both exposure and in pharmacology. And of course, if you haven't got evidence of those pillars, then you're less likely to succeed. And the next slide, hopefully, shows us the data. So we reviewed 45 compounds, which we put into the clinic over the last 5 to 10 years. And the teams that had kind of done their pillaring, as we call it, well, they'd kind of got those evidence of all those 3 attributes for their drug. Of the 15 teams that had that data, 12 of them actually achieved a positive POC, which is pretty good going, I would say. Conversely, the teams where they didn't have that evidence, and this is, you have to be objective about this, so maybe it's not the team you want to do that analysis. Of those 12, none of them achieved positive POC. And then there's some data in the other quadrant. So this kind of has changed the philosophy at Pfizer. Okay, every team now that's kind of going to a POC, they get asked by people like me and other leaders in the organisation, what about this stuff? Because it doesn't guarantee you success, but if you haven't got it, you're likely to fail. So biomarkers, I'm not going to dwell on biomarkers because others are going to talk about it today, but this is easy to say and not so easy to do. And the problem with biomarkers is often they're only validated after the fact, so you can only use them for the second or the third or the fourth compound. That's kind of my opinion. So, but it's still a great aim, because actually where you have translational biomarkers, they really help you, and we've seen evidence, and we talked about this morning in the pain area, and I think there were examples yesterday as well. So, quantitative clinical pharmacology, last 5 minutes. So, it's about, here's our business of moving things along, and what we want to do is have higher confidence early in the development process. And there are all sorts of clever ways which we believe will help us do that. And it goes through right to the real basics, but also incredible complexity, systems of biology approach, where you basically try and model the whole system in a computer. Now, at the moment, we haven't actually got one cell modelled in a computer yet, but it's a great goal. Where we are, I guess, in our world in Clin Pharm is kind of perhaps taking those systems-based approaches, those kind of approaches to deconstructing the system to the relevant pieces, and actually using sort of a systems-based approach to understand the pharmacology, which can be translated into some kind of PK/PD understanding, which then we can use to predict kind of doses and outcome. And so you've got this continuum of getting the right target and the right molecule, and then our business, I guess, ultimately in Clin Pharm is about getting the right dose. Clearly, we also need to get the right patients, so clinicians need to identify the appropriate patients, and I think that was talked about this morning in the pain session. So it's about translation. I'll skip this. And of course, what we tend to do is collect lots of data, and Donald Rumsfeld was the famous unknown unknowns, but these are kind of engineering terms in many ways. Actually, if only we knew what we don't know. And it's taking all that data and turning it into something informative, i.e. knowledge. So we collect all this stuff, and we all fill our IBs up with lots of data, but we tend to just throw them all in the IB, don't we? We kind of go, okay, we've got some preclinical data, we put that in section 3, and we put the toxicology in section 4, we put the clinical stuff in section 6, and we just list all these numbers, but we don't kind of integrate them. And Adam Cohen, who's the head of the CHDR in Leiden, was talking a couple of weeks ago at a meeting there about our business and what we do. We collect lots of data, very good, we're a very data-driven kind of industry. What we're less good at is kind of making sense of it, i.e. collating that data. And so our belief is that models, mathematical models, actually help us integrate all these data. And here's an example. I've got a few of them, which I might skip over some of them, but here's a whole bunch of studies, and it doesn't matter, you can't see them, but these are all a whole bunch of studies for some PD4 inhibitors. And actually, you can combine them. And actually, I think Jerome yesterday kind of had some nice examples, and Megan as well. This is basically our understanding of this is PD4 dose over its ED50, and then its effect on FEV1. So it's a kind of anti-inflammatory effect in COPD. And actually, you get some very consistent messaging. So you can go to literature, you can get information, and combine that in a simple form into a mathematical model. This is something I've kind of been involved with over the years. This is an approach we used on Maraviroc, which is a new treatment for HIV, where we have a pretty complex model, it has to be said. We've got a PK model, which is quite straightforward, a PD model, which is actually relatively straightforward. We use kind of Emacs type models. We have a disease model, which is based on viral dynamics, which is a kind of proper biological model from Bonhoeffer et al., and where we actually the drug concentrations influence the dynamics of the virus. And we can plug all that together and make some useful predictions. So here's a prediction of, this is kind of the maiden, really potent and successful anti-HIV regimen, efavirenz and two nucleosides. And based on that model, we can kind of show what the likely successes. So this is the number of copies of the virus, and you can make some predictions. From that, we actually made some predictions about the dose. So what you see here is for different Maraviroc doses, the likely success for different regimens. And basically, I've just put this up to show actually, the clinical team probably would have been down here if we hadn't done this modelling. But actually, the modelling said if you want kind of efavirenz-like efficacy, you need to be kind of around 80%, you need to be up here. You need to be doses above 150, 300 BD. Approved dose of Maraviroc without P450 inhibitors is 300 BD. So the model helps us. Coming back to some of the basic clinical pharmacology, drug interactions, biopharmaceutics. If you're running a food effect study or a drug interaction study, and you're a CRO or a physician or a study manager or whatever, the question you should ask your clin pharm colleague is, what are your predictions? Because everyone is able to do predictions of drug interactions and the biopharmaceutic properties now, today. Software is available, you should use it. I did this a few years ago, but this is Maraviroc, and we did a prediction in the computer, and we ran a clinical study. So here's a prediction, a ketocon's interaction. We predicted 4.5, we measured 5. Another study we did, we predicted 3.3. I ran a study, got 3. The third one, actually, we said, you know, I think we're convinced now. We did a prediction. We haven't run the study. We're confident, actually, at this point in this life, the life of this drug, that actually that's good enough. We'll probably run the study if it gets in the market in phase 3, because you'll need to have it on the label, but those predictions are good enough for us. Biopharmaceutics, this is food effect. I'm less convinced about food effects and our ability to prediction, but certainly in terms of predicting influence of dose and how that affects bioavailability, we can do those predictions. I'll skip that one for the sake of time. And I think this was talked about yesterday, so I'll go over this really quickly, but you know, we all know that phase 2A, we should all know that phase 2A is not simply small phase 3, and we can use novel, innovative designs to help us make decisions with less money. And so, for example, here's a pain case where the traditional endpoint is a 4-week endpoint, and we looked at data collected 2 weeks, because if we're running a study for 4 weeks, we always collect data at 2 weeks, and probably 1 week and 3 weeks as well. Actually, there's a really tight correlation. So run the study at 2 weeks. You don't need to run it for 4 weeks. Now you've just saved yourself some time. Again, and I think this was talked about yesterday as well, longitudinal models. Yeah, I'm nearly done. Again, we tend to statistically focus on the end of treatment and then throw away all of the data. If you take a longitudinal approach where you incorporate all the data throughout the study for week 1, 2, 4, 8, however long you're in, you can actually really save in terms of sample size. You can drop your, in this case, in this model or this analysis, 220 down to 128 patients, a massive saving. And if you don't believe models, I got this off the Met Office website last night. So this is a prediction for where all that ash is coming out of Iceland for 12 o'clock today, okay? This is a model. This is what, I guess, the people who run airports look at and make a decision whether your airport is open or not. So if you're flying to Europe, happily, today, hopefully, your planes will be flying because the model says the wind's blowing that direction, and I guess all the ash is going there. But these are models. They're not data, although they do fly planes through those clouds to just confirm that models are recovering what they measure. This is how other industries do their business. I was going to show you a thing about the space shuttle, but we haven't run out of time. So, you know, many of the other industries are quite comfortable using these model-based approaches. Now, there's always health warnings on models, and they've put theirs there, but you know, we can do the same in our industry. So hopefully what I've shown you is actually the challenges of clinical pharmacology. We are actually able to at least address some of those issues around our industry, and actually the quantitative approach that I've focused on actually can help us make our process much more efficient and make the decision making more speedily. Thank you.